Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Quick note before we begin, the Finding Genius Foundation, as part of the Finding Genius Podcast, has recently completed a book about understanding viruses. So the creation of this book was to interview 100 virologists, ask them a lot of deep, difficult questions, take the most difficult questions, and then re-interview the top 25 or so and ask them the hardest questions I could think of. And we compiled that all into a book. So you'll see question and 4 or 5 experts' answers. Question, 4 or 5 experts' answers. There's about 30 questions in the book. I think it's a great read for the layperson and for the researcher. It talks about a lot of speculation in the world of viruses, such as are they alive or not, and why is it important? Why do viruses go latent or hidden or ineffective or sit in a person or an animal or another creature for weeks, months, years, and then suddenly become virulent and affect that person? So there's a lot of really provocative questions in the book. It's now on Amazon. So if you go to Amazon and type in Finding Genius, you'll see the book on viruses. It's also on Kindle. The Audible version is in production and should be ready in approximately a month. But if you want to go and order it now, you can do so again by going to Amazon or Kindle, or go to findinggeniusfoundation.org and go to publications. There's an opportunity as well to get the transcripts of all the interviews and to hear the original interviews themselves. If we put them all together, the book would be about 1,000 pages, but we condensed them down to make it juicy and concise and tight and very interesting. So I hope you'll check out the book. We're now working on one about cancer, but this is going to be our goal is 3 times a year to come out with these masterclass books that I think will inspire new scientific research, and I hope you'll check it out. Thank you.
Hello, this is Richard Jacobs with the Finding Genius Podcast, now a part of the Finding Genius Foundation. Today I have Ravinash Krishna Kumar. He's a postdoctoral fellow at University of Oxford, and he's researching E. coli and how to use them to develop what's called 3D bacterial printing. It's part of the Hague and Bailey lab. So, Ravinash, thanks for coming.
Thank you very much for having me here. Pleasure to speak.
Yeah, if you would, tell me about your research.
Yeah, so this work's actually a cross between 2 labs at the University of Oxford here, in Professor Hague and Bailey's lab and Professor Kevin Foster's lab, who's based in the Department of Biochemistry and Zoology. And the idea for this work that started was to be able to adapt a 3D printing technology to position clusters of bacteria in defined spatial patterns to see how if we could print different populations and different strains and different species, could we print kind of community-based systems to ask and engineer communities for our own goal, to look at understanding how the spatial positioning of different microbes within these communities affects kind of the stability of them and the productivity of the community. So yeah, that's
what are you talking about?
Research.
What do you mean? You're taking E. coli and you're trying to put them into a stencil and just have them grow in a certain pattern?
Yeah, essentially that's right. So we use a 3D printer to print clusters of populations of E. coli into different patterns. So essentially the kind of the ground where they're stenciled into is a hydrogel, a very small little hydrogel of agarose, where we embed these bacteria within them, and we have different patterns of them, and then we see how they interact over time.
So what's the point of that? Are you trying to mimic how they look in biofilms, or what are you trying to figure out from this?
Yeah, it's a very good question. So it's thought of how in, say, like biofilm communities or any kind of microbial communities, whether they're living inside us in health, in agriculture, industry, or water treatment kind of microbial communities, they can often be, the species and strain can either be really, really mixed together, or they can be quite segregated in their own little patches. So a species might have like its little space in the community, and then some species will be patterned nicely around it at different scales, or they could be really intermingled together. So the idea was to use this 3D printing technology to kind of look at these questions. So if you have different strains mixed together, how do they interact? And if you have them segregated from each other, how do they interact? And the importance is the scale of interactions. So I think traditionally a lot of work originally done on agar plates would be kind of plating these strains on centimeter scales. But actually you would find these bacteria naturally patterned at micrometer scales, ranging from like 10 to like 1 millimeter kind of scales with these patterns. And this is what we can use the printer to do. So we can pattern them in these different scales and then look at how they interact at time.
Are they free to move, or are they stuck? How do they interact? What do you mean?
Yeah, exactly. That's a good question. So they are free to move, so they can move within this little agarose hydrogel, but they can interact in a lot of different ways. So they can interact by consuming local nutrients or sending out diffusible molecules to each other, or touching each other literally and sending things across. So yeah, I guess the question is what kind of interactions are we interested in? And the first ones we looked at was kind of looking at competition between these bacteria. So we picked E. coli because it's a very known bacterium, and it has this very strong interaction where it can produce very specific toxins that kill susceptible strains of itself. So these are bactericins, which are a type of antibiotic, and they can release these toxins to kill susceptible cells that can basically how they establish themselves within a community. So we wanted to look at kind of this idea of diffusible toxin interactions and spatial structure, and use the printer to ask these kind of questions.
Why would they release toxins if they're in a community that's not really community minded? If they're competing, I can see that, but it doesn't make any sense.
Yeah, I mean, I guess that's kind of describing how these communities are. So I guess a community in this sense doesn't matter whether it competes or cooperates. It's just them interacting in a certain space. And you're right. Yeah, there are instances of where the different strains of species will cooperate with each other, but there's also a plethora of examples where they'll be competing for space and nutrients within the community itself.
Well, if they're free to move, do you see a rearrangement to a particular pattern, or it sounds like a differential equation, and you have different starting conditions?
Yeah,
but the equation, if the species are the same or the strains are the same, it's kind of the same creatures there. What do you see a rearrangement, or what do you observe?
Yeah, so for us, if we're looking at this kind of toxin-based competition, we have the samples move a little bit, but they mainly just move by growing. So instead of like swimming around within the sample, they're just growing in their space. And you're right that these could be modeled really nicely via differential equations. But it's hard to test these kind of systems experimentally, hence the 3D printer. And what we're looking at is things not being there. So let's say, for instance, you print a producer and a susceptible cell completely mixed, and you fluorescently label each cell, and you look at the microscope in real time, them growing up, and then over time you'll see the susceptible cell literally disappear because it's been killed by the diffusible toxin. And then we can look at these different patterns, so printing them mixed together or kind of separated apart, to see how they interact. So yeah, if you're dead, you're kind of not there. You disappear.
It actually sounds like the game of Go. No, serious. I mean, I wonder if there's a line of bacteria or a region that grows into another region and changes it over for its own use. I mean, literally, it sounds like the game of Go. It's weird.
So yeah, I guess it's kind of playing these games with them. So I think there's been a lot of theory and larger scale experimental work. Is the idea of who's going to win in these situations when you have these two populations of E. coli competing. So if you generally
what do you observe that's useful? What are some things you've observed?
So we wanted to look at where coexistence between these strains happens, and kind of where also one strain would beat the other strain. And we're interested in coexistence because it's interesting to see how these competing communities can develop and sustain themselves. And I guess the idea of if you have dominance of one strain, this is related to possibly how a pathogen might come in and break down a community. So yeah, diversity is key for this kind of idea. And so yeah, we're interested in looking at how they're mixed and segregated with each other. So the kind
Well,
of
do you see that they form biofilms, or like do you see certain strains never get along, or certain strain variants, or for some reason they just take over everything?
Before we continue, I've been personally funding the Finding Genius podcast for 4.5 years now, which has led to 2,700 plus interviews of clinicians, researchers, scientists, CEOs, and other amazing people who are working to advance science and improve our lives and our world. Even though this podcast gets 100,000 plus downloads a month, we need your help to reach hundreds of thousands more worldwide. Please visit findinggeniuspodcast.com and click on Support Us. We have 3 levels of membership from $10 to $49 a month, including perks such as the ability to see ahead in our interview calendar and ask questions of upcoming guests, transcripts of podcasts you're interested in, the ability to request specific topics or guests, and more. Visit findinggeniuspodcast.com and click Support Us today. Now back to the show.
Yes, that's right. So the E. coli strains we were looking at, the producer produces this toxin, and you get different types of toxins. So the toxins work by kind of really interesting. So a producer cell has to lyse and release the toxin. So it's kind of sacrificing itself to protect its clomates. And basally about 1 in 200 cells will lyse and release this toxin. And the clomates aren't affected because they also encode for an immunity protein, which neutralizes the toxin. So you're not going to self-intoxicate. And when this toxin is released and diffuses, it will translocate into the cytosol of the susceptible strain. And will go in and either form holes in the membrane or break down via DNA damage or break down via RNA damage as well. So, and the difference between these two strains are literally one's carrying the plasmid that makes this toxin, and the other strain doesn't have it. So they're almost identical but not quite, just because of this difference of carrying the plasmid. And the idea is that we found as you might predict, when they're mixed together, that the producer always dominates because it has the increased chance of interacting with each other. But when you start separating them apart from each other, you get scenarios where the producer completely still wipes out the susceptible. But we found really interesting scenarios where you have kind of weaker toxins that the susceptible actually outcompete the producer, and that's kind of interesting because I guess we didn't expect it on these very small micrometer scales. As we know, these toxins can diffuse over really large scales and kill on plates.
Well, do you perturb the system? Do you heat up, a part of the plate where everyone's hanging out, or change the pH in one corner of it, see how it affects all the bacteria across the plate?
So yeah, not at the moment. So yeah, the nutrients are completely homogeneous in the system. They're all exposed to the same thing. It's just the imposed pattern. So where they start with each other, and then you watch them on the microscope, kind of fighting over time.
Why not perturb one of them and see what happens? Make one area of the agar a little bit inhospitable and see if they migrate or what happens then.
Yeah, I mean, this is really interesting questions you're asking. So yeah, with the printers, the idea that we could print in already these perturbations, so you could have certain gradients of nutrients, or like you said, lowering the pH in a certain area, and see how these bacteria move between them. But the idea for us
it'd be weird if you made a cache or a cache or whatever, a bunch of nutrient in one area and no nutrient around it, and see if they race towards it and compete to capture the flag.
Yeah, exactly, and see how they kind of capture the flag. That's right. But the idea is for us is to use this printer as a way to test this kind of spatial relationship. So if you've got a hypothesis about spatial structure between these strains, of how they're patterned, should they be mixed together to coexist, or should they be separated to coexist? We can now look at that and test it.
Are you doing a 2D gel or a 3D? Because from what I understand, biofilms obviously are 3-dimensional. In nature. And do these things form biofilms, or do they seem to act separately on the agar?
Yeah, that's right. So initially we started with 2D patterns. So the hydrogel block that we print is about 900 micrometers by 900 micrometers by 100 micrometers. So that's in the X, Y, and then Z dimension. So it's a very thin little square block. The patterning we've done is initially 2D to start with, as we thought this was the easiest way to go, is to look at how if you separate them or mix them together in different patterns, But you're right, it's very interesting if we start looking at 3D patterns with different nutrient gradients on the top and bottom of how they develop over time. And at the moment, yeah, so these bacteria start from single cells embedded within the agros, and then they grow into these microcolonies.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
Which interact via diffusion, so nutrients and other secreted molecules, but they're not completely a homogeneous kind of biofilm as you might see when things grow on the surface. And these are more reminiscent of possibly like how different bacteria grow in deep tissue wounds, or even possibly in the part of the mucus layer in the gut. So yeah, it would be interesting to look at these in 3D as well.
Well, what kind of things are you testing? What kind of patterns? Like, are you imagining in the agar, you make a mark as if it was someone's skin that was cut, maybe a 200 micron cut in the agar where there's no nutrients, and see what the bacteria do as if they were, entering a wound. I don't know what kind of geometries would be useful for you to study and why.
Yeah, so yeah, I'll go into this now. So initially with the idea of being mixed or segregated, we can do graded degrees, which is the simplest start. So if you imagine starting for like you mixed genotype NA and B together, and there's kind of no spatial structures, so you see lots of like blue and green kind of community little colonies within this block, and then you can start creating more patchier in a checkerboard fashion. So you might have small little blocks of 50 micrometers where you have green and red, then 100 micrometers, and then 200 micrometers, and then to the point that your block is just you've got green on one side and you've got red on the other, and then you can look at the degree of the mixing between these interactions. So other patterns we looked at is also to probe the biology of these systems as well. So the idea when you have a producer on one side of the block and a susceptible on the other side, we had scenarios where the susceptible would completely outcompete the producer, and we had this hypothesis that the producer is producing this toxin at this interface, and we thought that the clomates at the interface of the producer susceptible strains were absorbing the toxins so that other susceptible strains grow behind and proliferate really well. And the way we proved that is by producing strips of different genotypes. So we had a strip of a toxin producer, and then strips of different susceptible strains, and we could change out at the interface what strains we put there. What we worked out is that susceptible strains were actively absorbing the toxin to protect other strains from behind, and let them grow up. And we found it was really due to having this receptor that the toxin binds to. So if you deleted the receptor, the toxin would just diffuse straight through and kill everything at the back. Whereas if you had this receptor in the susceptible strip, it would take it up, absorb it, and protect strains behind it. So it's interesting, like by probing kind of these fundamental questions of space structure, you also kind of elucidate biological mechanisms that you weren't really aware with to start with, that you could hypothesize that could happen.
What do you mean they're picking up? I don't understand. So there's a leading edge of once bacteria have grown into a microcolony, the leading edge of them will pick up what useful molecules and then spread it to the bacteria that are within the microcolony behind them.
So yeah, not quite. Sorry, so I don't think I've explained this well. So say you have lots of colonies of this producer strain, and then next to them you have lots of colonies of this susceptible strain, and there's an interface between them, so they're divided up nicely. So you have producer on one side and you have susceptible on the other side. So the producer is going to make this toxin that will diffuse from the producer's colonies and then start hitting the susceptible microcolonies. And this will start killing the susceptible strains. And what we found is that the susceptible strains nearest the interface they were dying, but they were also grabbing lots of other toxins in the process. So they were like diluting up the concentration of toxin, so that the toxin didn't get past them to other susceptible strains behind them to let them grow up. So it shows the importance of structures in these systems. And
so they were sacrificing themselves, you mean?
Yeah, well, yeah. One could say that they are sacrificing themselves to do it. Whether this is just accidental sacrifice or some kind of evolved mechanism is yet to be determined, but it definitely happens. And what's really cool is about the print is that, you have this ability to kind of, make these nice little strips to test these examples. So say you put A in front of B or B in front of A, you can see if they absorb the toxin or don't absorb the toxin. And then you can make mutants to see what receptors are responsible and what receptors, stop this toxin going past. So it's kind of like seeing this, I guess, synergy between what might be traditionally 3D bioprinting and kind of looking at the ecology of the system, which is really interesting.
Well, again, looking at these microcolonies, are they bound together? Are they still free floating? Are they biofilms? What do they look like?
Yeah, so these microcolonies are, they grow from single cell dispersions into like 3D spheres that are about, depending on how tightly you pack them, about 30 to 50 micrometers in kind of radii of these spheres, and they're tightly packed spheres of bacteria that have also these kind of EPS that are produced between them. So I think people are starting to look at the 3D structures of these microcolonies, especially when it comes to looking at diseases of these microcolonies in tissues.
And what is EPS?
EPS is external polysaccharides. So bacteria, when they're forming biofilms, can produce these polysaccharides and polymers to protect themselves from the environment, essentially, and to stop things.
Well, what do you observe within each colony as it starts growing? It starts from a single bacteria, I guess, and then each of the microcolonies, before they interact, what do you notice on how they grow? What's interesting about them?
So they kind of, I think they kind of grow as you might expect. So like if you look at them on a 2D plate, they kind of grow in a nice monolayer. So they're just doubling. All these bacteria are doubling around each other, and then they start stacking on top of each other. So if you say you embed a bacteria in a hydrogel and you look at the grow over the time, this is kind of replicated but in 3D. So you see, it divide and divide again, divide again, divide again, divide again. And then if you stain for these certain polymers, you can see them starting to be produced over time. So that in itself isn't, I would say, like, really novel or anything like that. But what is interesting is that I guess you can look at, see these toxins coming into the system and hitting these kind of colonies and seeing how they develop over time. And the idea is that, you see these ones at the edges of the colony are kind of absorbing it and protecting the ones in the center of it.
Well, what if you have two colonies separated by a number of bridges instead of just, they can connect to any way they want, and the toxin has to come across a bridge or two? It might show you different behavior about how they interact. Again, if there's only, one or two ways in for the toxin or interaction to happen.
So yeah, the toxins are just diffusing through this hydrogel.
But again, they don't have to. You could put impermeable layers or impermeable barriers so that it can only go through one channel.
Yeah, that's right. You could do that as well and see. In a sense, this is what the bacteria are doing for their clomates. They're creating those kind of impermeable channels or protective barriers to let their clomates grow up. So yeah, I think the system is amenable to a lot of these different changes when it comes to kind of looking at spatial mixing and spatial segregation. I think where we want to go with this a lot more is build a lot more complex kind of communities. So the example given here is looking at these two competing populations of E. coli and looking at how if you mix them together or segregate them, what if one wins or one loses, and what kind of mechanisms to keep one winning or losing. But if we start printing lots of different strains and species, it'll be interesting to see how spatial structure has an impact on their interactions, whether they're mixed or segregated in these patches.
Well, what about critical mass? If you have a colony how many bacteria form in these colonies typically before they turn on a certain activity? Is there quorum sensing based behavior that doesn't occur until the colony has 1,000 bacteria in it or something?
Yeah, that's a really interesting question. So these toxin producing bacteria, these toxin producing E. coli that make the colicin, some strains that we've used in our experiments do have this ability to reach quorum and upregulate the toxin production. So the idea here being that if you're a producing cell that's lysed, and then the toxin diffuses around and it hits another clomate, they might import the toxin and then they'll amplify their toxin production. So you can see these kind of progressive ways at the interface of when one toxin producer lyses, another one will lyse and so forth. And then in these examples, you see that massively amplified when you say you have two producers against each other. So the way that the toxin is released is dependent on DNA damage. So if you have two producers against each other that, are both creating DNA damaging toxins, they're going to both massively amplify their toxin production and annihilate another, which is really interesting. So with regards back to the question of spatial structure, when we put these two auto-inducing populations of E. coli so they can amplify their production of toxin in some kind of sensing mechanism, we found the more dominant when you mix them together would win. But really interesting when you separate them together, which is, the prediction is usually when you separate things, you'd expect coexistence because they interact more weakly. We actually found, mutual destruction of both of them. And the idea here being is you give each other an ability to build up the toxin and then send it across to one another, and then they completely eliminate each other. So sometimes the predictions that seem intuitive aren't always the ones that happen.
What about phage activity? How are you supposed to reckon with that? What if a phage is killing one of the strains and it's not the toxins themselves?
So yeah, we haven't really worked with phages, but I think looking at phage,
well, you probably are working with them unintentionally because they're probably coming along for the ride.
So I mean, yeah, in E. coli they will probably have some cryptic phages, meaning that they've got some genes, I guess, from phage that don't make the phage anymore. But in the toxin system itself, there aren't any phages involved that we know of. But yeah, I think looking at like phage progression through these systems would be really interesting as well. So again, if you have clomates that can that are resistant to phage, can they protect phages behind and so forth? But yeah, I think that is very interesting as well.
Well, I don't know how carefully you could filter, but if you're not filtering down to, I guess, the 50 or 100 nanometer level, I don't even know if you could do that. You probably have phage coming along with you that are in the gel with the the bacteria. They're not probably alone, would be my guess.
If I accidentally sneeze on the sample or something as well, then you could have a lot of different phages. But yeah, within the system, the phages will easily diffuse. So say you printed phages and wanted to see how they propagate through different bacteria barriers, on one side of the hydrogel array, they would easily propagate, go in, lyse the bacteria and then carry on propagating through the system. So it'd be interesting to see how to stop these kind of progressions. And these have been reported, naturally in biofilms as well, right? Seeing it.
So again, what are your goals? What are you trying to figure out specifically?
So yeah, the current goals are to just generally look at what kind of structures in a microbial community can make the community productive. Productive being increasing its biomass, or stable, meaning if you perturb it to using an antibiotic or pathogen, how quickly it can recover. So the idea is to print lots of different strains in either mixed or these kind of segregated clone patches to see what leads to kind of coexistence over time. And we did this test, with E. coli because they're very well studied, and this colicin system is known in E. coli because they naturally compete in our gut and in other guts, using these toxins to establish themselves in space. But to progress it is to look at different kind of bacteria that we find within us. So to look at a subset of say very dominant species within our gut microbiome. If we print them, mixed together, how do they interact, and does one strain dominate over time, or do many strains stay diverse? So that's kind of where we're going with the technology and what we want to look at.
Yeah, why do you have only E. coli? Why don't you try to mix E. coli with one species and then E. coli with another one and another one to see the interactions and to elucidate possible interactions in the gut, for instance? Yeah.
That's exactly what we want to do. So it was just a test initially with the E. coli. So we're currently now working with the genus Bacteroides, which is one of the most dominant genus, I guess in our gut. And we're looking at maybe 5 to 6 different species that are known to interact within our gut, and to print these in different patterns of being mixed and segregated to look at how they develop over time. What's really interesting is between it, as we were talking at the beginning, this idea of competition and cooperation between these interacting species, is that the Bacteroides, are known to kind of to cooperate with each other within the gut microbiome, and also compete. So they're kind of known as the foragers. So they can produce a lot of these extracellular vesicles to degrade certain food particles that we can't. At the same time, they can also produce a plethora of toxins to kill one another as well. So we know that they're very dominant. And they exist. There's not necessarily a dominant species. They exist. Many species exist within us. So it'd be really interesting to put these strains together and species together and see, does one win? Does one beat all of them, or are we going to get a stable kind of ecosystem?
Well, can you try one or two experiments where you get some gut cells from a person, keep them cultured, and then put different kinds of E. coli strains in there with them and segregate them and put them together and see what happens?
Yeah. These are all really good questions. So I guess this is more about the technology. So initially, we just wanted to test this kind of use this 3D printing technology for just looking purely about bacteria and asking these kind of fundamental questions of space and interactions. But looking at, can we develop it further? So yeah, you're right. Can we print some of these, gut epithelial cells to, create a frame and then have a really nice kind of mucus layer on top where these bacteria would sit, and then, yeah, exactly that, print them in these different patterns on it to kind of see, is there any kind of host interactions? Another way is we would like to look at is kind of pathogen evasion assays. So say you kind of print a small community of strains, and then you can just put you can print in a pathogen, and you can see how it propagates throughout the community. And depending where you put the pathogen, it might be interesting to see if certain strains are needed to outcompete this pathogen.
Okay, well, very good. What's the best way for people to find out more about your research? And I don't know if you have any cool pictures. And I don't know if you spelled out your name in bacteria, like the Ravinash colony fights the Susan colony. That'd be funny to do.
Yeah, I always, at the end of my talks, I've got a smiley bacteria printed face that I always put in. But yeah, you can find out more about this research. If you look at the Hagen-Bailey group and Kevin Foster group websites, and we recently published some papers on this that you can find out about it, which is really cool.
Very good. Well, Ravinash, thank you for coming on the podcast. I appreciate it.
Yeah, thank you very much for having me. It was a pleasure to talk.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.